Before this experiment, one screw cap and one flip-top test tube for each of the muscle samples was labeled. In this case, only one is being studied. Following that, add 250 macroliters of Biorad Liamli sample buffer to the flip-top test tube using a pipette. Then, transfer a small sample of muscle into the flip-top. Next, seal the tube and agitate the flip-top test tube by flicking it repeatedly, up to 15 times. Then, incubate the sample at room temperature for 5 minutes. Following that, transfer the buffer from the flip-top to the screw cap. Make sure that you do not transfer any of the flesh when doing this step, only transfer the buffer. Tightly seal the screw cap, then place it in a foam test tube rack, making sure the buffer is completely below the foam, and then place it in the water bath for 5 minutes. The water bath has previously been heated to 95 degrees Celsius. When the 5 minutes are up, carefully remove the screw cap and the foam test tube rack from the hot bath and bring it over to your table. Then, prepare a gel cassette by placing the gel into the electrode assembly with the short plate facing inwards and then placing a buffer dam on the other side. Clamp the assembly together and place it into the electrode chamber. The gel will be the medium in which proteins will be separated and analyzed. The next step is to fill each well of the cassette with the appropriate sample using a micropipette, starting at well number 3. Wells 3 to 9 will be injected with 5 macroliters of the flesh sample from the screw cap. But before pipetting anything, you must ensure that the micropipette is properly set to the amount you need. This is done by adjusting the top. In this situation, it has been adjusted to 5 macroliters for wells 3 to 9. After these wells have been filled, well 10 will be injected with 10 macroliters of actin and myosin standard. Before pipetting the actin and myosin standard, you must change the tip and remember to readjust the micropipette for well 10 to 10 macroliters instead of the 5 macroliters used for the previous 3 to 9 wells. Once the micropipetting step is finished, place the lid on the tank and connect the leads. Then set the voltage to 200 and let it run for 30 minutes. When the gels are finished, make sure to turn off the machine and unplug the leads. Remove the electrode assembly from the chamber and take out your gel cassette. Carefully pry apart the plate and then transfer the gel to the tray and gently rinse with water. Then add BioSafe Coumasin stain to the clear gel and let it sit in the stain for 60 minutes, shaking gently periodically. The gel should stain a deep blue. This step is easier to do when lab partners take shifts throughout the 1 hour. Then transfer the gel into a container that is filled 3/4 with deionized water. You must be careful not to break the gel when transferring it into the container. Place the container in the microwave for 45 minutes. When the time is up, remove the container carefully from the microwave and let it cool for 10 minutes. When cool, transfer the gel back into your tray and place it in an electric scanner. The electric scanner will detect the bands on the gel that indicate a protein. Finally, interpret the results obtained from the gel scan.